Well, good afternoon, everyone. Wow, what a great crowd. For those who don't know me, my name is Bill Ermatinger, and as Barry so graciously introduced me earlier, I am the Executive Vice President and Chief Human Resource Officer for Huntington Ingalls. There's a mouthful. And as in tradition, past chairs of Barry appear to always kind of migrate to be then the current chair for the Virginia Early Childhood Foundation, and I'm so pleased to be that role. First, the VCF and the Chamber, we are extremely fortunate that we have combined forces. This is our fifth year, and I think it speaks volumes on how important this connection is with the business community and with early childhood ed. We always talk about workforce development. Barry just got back from doing Blueprint 2025, and everywhere he went was workforce development, workforce development, workforce development. As an engineering company, you've probably heard me say this before, anytime you have a problem, engineers typically go back to, okay, what's the design? If this thing broke, what's the design? Let's pull out the design first. That's what an engineering does. And so when you look at workforce development, we go all the way back to the design of workforce development, which is basically birth, and in some cases pre-birth. And so this connectivity to workforce development and early ed is so critical. I think we all understand even more clearly why Virginia needs to focus on this attention and the resources on ensuring sound policies and programs for our youngest citizens, which brings us to our first speaker, Dr. Michael Freelander. As you can see from his bio, Dr. Freelander has an impressive set of credentials. He joined Virginia Tech Carleon Research Institute, its founding executive director in 2010. In 2016, he was appointed as the Vice President for Health Sciences and Technology at Virginia Tech. His research focuses on the processes that underlie learning and visual processing in the brain during development and after traumatic brain injury. His work gives us important insights into how to best support the workforce pipeline from the very beginning, and touches on how the earliest days, weeks, and years of life can shape a lifetime. We're extremely fortunate to have him here today. He travels around the world, and we are very grateful and thankful that you joined us today. Please welcome Dr. Freeman.
Thank you. Pleasure to be here. Can you hear me okay? All right. Okay. My talk is probably a little different than some of the others you've had if you looked at the title. I'm going to talk a little bit about biology. I want everybody here to appreciate we're all biological organisms. We live in that world, so do our kids and grandkids. So what I am going to say to you hopefully will connect to policy and so forth, but from a biological perspective. Does everybody know what that is? Okay, good. We're off to a good start. From my perspective, that is one of the most important, if not the most important thing in the universe. It's a source of creativity, it's a source of inspiration, it's a source of building. It's also the source of some things we'd rather forget probably, but it's the source of incredible power. The human brain is a phenomenal computational machine, and putting that together requires a lot of things to go right. For those of us who study the development, the very fact that it ever comes out right is amazing, because of all the complexities and the places you think it might go wrong. These are some of the players that live inside that organ. These are called nerve cells or neurons. I don't know how well you can see it, but they're studded with little bumps on them. And those little bumps are called spines, highlighted in that. And each nerve cell is a site of communication from another synapse. Many of you have heard these numbers before. You got lots and lots of nerve cells in your brain, and then each nerve cell has lots and lots of synapses, and when you multiply them, you got a big, I don't even know what you call it, quadrillion or something like that. It's a very powerful computer, runs on 20 watts of energy, much more efficient than anything else. That's important, and it's surprising in many cases. This is a picture taken at a high level of magnification of one of those connection points called a synapse. It is the site where one nerve cell talks to another. These little circles you see are called vesicles that release chemicals, where nerve cell A says hello to nerve cell B. Those are the communication points and the points of modification where what we call synaptic plasticity can occur. Now let's think a little bit about who we are and where we fit in the animal kingdom. Let's just talk about mammals. I don't want to give you a whole biology 101 course. These animals on the left side here are called altricial, and these animals on the right are called precocial. And you probably are familiar with all of those. The horse, the colt on the right, is a precocial animal. Many of you probably seen a horse being born and see how they jump up and run around. It's amazing. You won't see a kid doing that. Turns out guinea pigs are like that as well. Quite precocial. Their brains are quite developed when they're born. On the other hand, this is a baby mouse, and you can tell just looking at it that it needs care. This is a baby monkey, a macaque monkey, and this, of course, is a baby human being. And we're much more like these than we are like these. And so we are born at a very early stage of development for our brain and require a lot of things to go right, not only in the womb, but after birth. And there's a very protracted period for learning to occur to make us into what we are. And it's those things that I want to talk to you about. This picture is a series of pictures going on through gestation of the human brain during development. You see how much going on. Cells are moving around. They're making those synapses connecting to each other. The wiring diagram is being laid down. And many things in the environment, the local environment in utero, the environment around us can affect that in dramatic ways. And again, I apologize. You probably can't read this, but suffice it to say, this is a time plot going all the way out to, say, 70 years old from conception, and showing how a lot of things develop and what they depend on. So what it says here is higher cognitive functions. You can see how long those take to develop. You see receptive language and speech here. Here you see sensory processes such as vision, hearing, and so forth. And then here you have a number of biological processes you probably can't read, but it says synaptogenesis, myelination, the things happening to red nerve cells and insulation and creating the synapses. So all these processes are going on for quite a long time in our brains. And this is what it looks like if you look inside the brain of a newborn and then 1 month old, 6 months old, a couple of years old, through a microscope and look at the nerve cells. You don't need to quantify this or need to tell you can see. There's quite an elaboration and growth. Things are changing and changing dramatically. And this little plot here shows what happens in terms of the number of these chemical communications between nerve cells, synapses. The numbers go up, and then they start to go down into the first couple of years of life. That's called pruning or elimination of synapses. So it's as if the circuit overproduces a lot of connections, figures out which ones are important, stabilizes them, and gets rid of the rest of the stuff. That's a simple look at it. But now let me tell you an uncomfortable truth. First, let me ask a question. Is anybody in this room over 25 years old? This is for you, all of you. Is anybody over 40? Okay. The uncomfortable truth is older brains are not so plastic. There's been a lot of hype the last 20 years in science and in the press about all the great things our older brains can do. And that's a whole other talk that I did where I talk about how exciting it is for older brains to learn new tricks. And we can. I don't want to give the wrong message here, but there's a fundamental truth, and this is the fundamental truth. Younger brains are much more plastic than older brains, no matter how you cut it. They're much more susceptible to modification. And so if we look at a variety of things such as the brain's ability to change in response to an experience, we can see that's a decreasing function. And then we look at what's called the amount of effort. This has been measured a variety of ways, energy expenditure, activity in the brain, etc. It takes more as we get older. So that's the truth. So for those of us over 25, okay, we'll struggle with that and we'll figure it out. But we're really talking about for the kids. And they are incredibly susceptible to intervention on the good and the bad side. Many of you have probably seen this picture. It's from the Romanian orphanage study that was done. And these are children that were kept in isolation in Romanian orphanages. They've been studied and followed up for many years. And let's just say the outcomes were very, very bad. And the consequences are virtually lifelong, even though all kinds of interventions have been tried. Those earliest experiences were formative for their brain and their behavior. That's kind of the dark side of what's going on. Now, some of you are probably old enough to remember this movie, The Graduate, where Mr. McGuire said, I want to say one word, just one word, and it says, yes, sir. Are you listening? Yes, I am. Plastics. Exactly what do you mean? There's a great future in plastics. Think about it, please. Yes, I will. Enough said, that's the deal. So this whole plastic thing has been around for a while, the idea that we can mold things. And there's a whole story there about our environment and the great things we've done with plastics and the challenges. But we think of brains that way too, as malleable, particularly for a period of time. Now this is where the uncomfortable truth started. This is right from my own field, and I'm very familiar with this work. This is a famous picture that led to a Nobel Prize for these two individuals, friends and colleagues of mine, David Hubel and Thorsten Wiesel. And they did some of the earliest studies to see what happens if your eyes aren't appropriately aligned when you're a child to your brain. And in a nutshell, this picture shows what the back of the brain should look like normally. The black and white stripes represent synaptic inputs coming from one eye and the other in an alternating fashion. This picture represents what the brain looks like if there's an imbalance, and it's dramatically shifted. And we got that idea very early on that there's an early period or a sensitive or a critical period from studying that when the brain is susceptible. I like to emphasize this particular study that was done quite a few years ago now. It's on studying folks that are learning how to play a string instrument, a violin. And again, I realize you probably can't see it, but what it says on the x-axis here is the age at inception of musical practice, starting with 5-year-olds up to 20-year-olds and then adults. And this is a measurement using a technique. The technique isn't important, but it's basically measuring the strength of activity in a part of the brain called the motor cortex that controls the spinal cord, that controls the fingers that the player is fretting with. And you can measure how effectively that part of the brain is working. And what you can see here is for the students who started violin playing at the earliest ages, very dramatic effect on the brain. For those who started, in this case, in teens, some effect, but not nearly what that was, and for the adult controls, even less. This is often used to tell people that there's still hope for adults. It's above zero. But I want to turn it around here and emphasize this plasticity that happens in children, the ability to learn things when you start early. And we all know that. You didn't need millions of dollars of science probably to tell you that, but we didn't know it precisely. So what happens in the brain during this period of development? We now know there's widespread migration or movement of nerve cells into the frontal lobe of the brain throughout postnatal life. All these green dots represent a type of nerve cell called an inhibitory nerve cell that are moving in the frontal poles of the brain during development. This was published a couple years ago, but it's only during the early years of postnatal development. This picture, which has a lot of stuff on it, is from a study that came out just a few months ago that's highly controversial and being talked about in science a lot. It basically debunks the idea we've held on to for the last 20 years that adult brains make new neurons, what's called neurogenesis. We were so excited when some of our colleagues discovered in the adult brain, there's some really interesting studies having to do with radioactivity from nuclear testing and from cancer patients in Scandinavia who receive radioactive materials that allow you to date after somebody dies when a cell was born or generated, so-called neurogenesis. And there were studies suggesting in the adult brain there was a lot of it. Well, it turns out a lot of that was an artifact. And we're now realizing that that neurogenesis is happening primarily in the earliest period of life only, not very much later. So making all those new cells and connections and synapses is an important part of what happens. And that leads to a functional change in the brain as indicated here. There's a picture of the human brain highlighted in blue as an area in the side called the temporal lobe. This is the hippocampus there. This is an electrical recording from a cell, and this is a phenomenon called LPP, long-term potentiation. What we're measuring is the size of the response of one neuron to another, which stays at baseline until there's strong activation. And the minute that's over, the second it's over, the synapse gets stronger and stays stronger. And this would go on for hours or days. In other words, brief, intense periods of activity can permanently change the strength of synaptic connections in the brain. Not only is the strength changed, but these connections literally jump around in your brain. I remember I had the great fortune to spend a day with a gentleman named Francis Crick, who many of you may have heard about of Watson and Crick fame for discovering the structure of DNA. He visited my lab when I was in Birmingham, Alabama, to talk about some work like this, and he had this crazy idea that our brains were twitching all the time. And I said, where did you get that idea from? He said, well, there's a molecule in our nerve cells called F-actin. That's what we have in muscles for contracting muscle. And there are all these little connections called spines and synapses. I think the way they're changing their activity is by moving. Everybody thought he was crazy. You know, a Nobel laureate came into a new field in neuroscience saying things that were probably wrong. Well, fast forward when the technology became available, fancy new microscopes called multiphoton systems, we looked at them and they're jumping around. He was absolutely right. Our brains are jumping around, and through that LTP process, these little spines and connections are pulling out, pushing out, making new connections all the time. Your brain feels like Jell-O, but it's a dynamic, living Jell-O. It's literally making and breaking physical connections. Speaking of making and breaking physical connections, I want to bring to your attention this study. I think many of the people in this room know Dr. Craig Rainey, Dr. Sharon Rainey, who work at our research institute. So Craig was the first person to start a study known as the Abecedarian Project, along with his colleagues at the University of North Carolina. And this was a very intensive intervention for children early on for health, nutrition, education, etc. Children at very high risk and otherwise very challenging circumstances. And those kids for the first few years of life did better on every metric you could measure. Fast forward, Craig and his group have now studied these people for 40 years. They come to Roanoke, Virginia on alternate Sundays for follow-up studies with their children and their grandchildren sometimes. And they participate in functional brain imaging studies. And working together with some of our other scientists, particularly this gentleman, Rainey, they've been doing functional brain imaging, and their study was just accepted this week in a journal called Nature. It's going to get a lot of visibility, and what they found was not only did all these benefits continue to accrue, but other health benefits such as cardiometabolic health, a lot of other things have happened and lasted for decades, and back to the brain and jumping around, the connections in their brain have literally changed. You can visualize it with functional brain imaging, and it has lasted a lifetime. 40 years from that very intensive early intervention. So this is real-world proof of what we're learning in the laboratory makes a difference. Now, who hasn't seen this situation with their kids or grandkids? And what do we usually do? Come over here, go outside, play baseball, put that down, whatever. These interactions with electronic devices are a challenge. We're running an experiment, probably the greatest experiment in the history of the world, what that means for development of the brain. This is a slightly different version. What's the difference between these two? Social interaction. This person is engaging with broadband communication with another individual, not just with a rather impoverished environment on an electronic device. Well, we do the same thing in the lab. We can work with animals who get to play with each other, or they're reared alone and so forth, and we find that there are great effects. So this is a study done again by Dr. Montague at the Virginia Tech Growing Research Institute, where we looked at group interactions and how they affect cognitive performance in individuals. There's a massive impact on brain function of group interactions. Isolation is basically not good. Of course, we knew that, but here's direct evidence of what it does for development of the brain. So whatever you do, working with devices and so forth, if you can integrate the broadband communication with other people, it's going to make it a more enriching experience. Now, a couple slides with a lot of data on them. I won't go through it. Let me just tell you what it says here. This says repetition causes efficient activation of learning-gated signals to facilitate long-term memory and lead to utilization of learned information. This is a kind of a molecular signaling pathway inside cells in a part of the brain where we know learning occurs. We now know, have lots of evidence that repetition actually changes the mechanism, gets right to the DNA, the genetic material, and changes the hardwiring of the nerve cells. So repetition is good to a degree. But this says space versus mass activation is more efficient at inducing biological substrates of memory during long-term information storage. In other words, repetition is good, but if you cram, that's not good. You have to space it out. Now, there's probably, I don't know if there are any teachers in the room, you all know that. I struggle constantly with development of curriculum in universities where people are making modules and cramming things into smaller blocks of time. If we can get all this in within 2 weeks, they can learn that and something else. That's not how brains work. You gotta have time to digest it, use it, put it to work, recalibrate it, and come back to it, revisit it internally. And these are biological experiments that show that makes a difference. Stress is another issue we worry about. This is that part of the brain called the hippocampus in the temporal lobe where initial memories are formed. The reason all these cells are black is they've been stained with an antibody for a stress hormone. And what that means is these cells in the brain where learning happens are particularly sensitive to stress levels in your body as you release hormones indicating stress. Now, we generally think stress is not good. Everybody's always trying to avoid stress. What I'm going to tell you in a moment is that a little bit of stress is okay. A lot of stress is not okay. Chronic stress on this circuit in the brain will absolutely destroy its ability to store and utilize new information. But, coming up here and being a little nervous, a little wired, or thinking about that exam, that's okay. You can be on your game, and you can focus your attention, and you can modulate through what are called neuromodulatory substances in the brain, the level of activation and utilized circuits. So as you think about constructing environments, curriculum, etc., don't completely shy away from high-stakes situations. You do want to shy away from where the individual has little to no control or it's a chronic stressful situation, and we can see that in the biology. And one of the last things I want to mention is this concept of learning by doing and learning by thinking together. This is a functional magnetic resonance imaging study. There are many other parts of the brain involved. This just happens to be looking at one part of the brain called the cerebellum. This is measuring the response you get. And the bottom line is, what is shown on the right here is when you're both meditating, thinking about what you're doing, in this case it was a writing task, and doing it, versus just thinking about it. And I had other slides I don't have time to show you where people do this with all languages, learning physical sports, etc. And so while you do things that you're thinking about, the circuits synergistically interact to reinforce your thinking about it. And so as you work with students in the classroom setting, wherever it might be, the more hands-on and involved they are, the more likely what you're teaching them in a more abstract way will be meaningful to the circuits that are likely to store this information and make it accessible for future utilization. And last kind of scientific point is this one. It's called memory replay during sleep. What you're looking at is the activity. Each little dot represents activity of a neuron recorded in that part of the brain that we were talking about before. And these are recorded from the brains of mice. Now, these mice learned their way around a maze during the day. They were finding their way around the maze, and the Nobel Prize was awarded last year for a discovery about how this affects what are called place fields in the brain. You make a map in your brain of where you've been, and you use that mental map. And these mice do that, and then at night, what happens is they replay the activity pattern. And it's the exact same pattern that happened when they were learning their way around. If you disrupt their sleep, the replay is disrupted and the information does not consolidate. i.e., rest is important. It enables what you've learned during a period of time to consolidate. We have hardcore biological information to show how that works. And I want to just wrap up by saying I'm going to cite the work of another one of our researchers, Dr. Warren Bickel. Dr. Bickel doesn't study development per se, he studies addiction and substance abuse. And he particularly studies something called temporal discounting, how we value the future and how we make decisions, whether it's a decision to take a drug or a decision to invest or bail out of the stock market, or whatever it might be, a health decision based on the weight of value we give to things in the future. And so he studies this, and he's found it as a disease process, and what he says is excessive discounting is putting our futures at risk. And these are some data. This shows the actual discounting function for a normal control. This is somebody who's addicted to opioids. So instead of a 4-year time curve, they've got a 9-day time curve. Their world ends in 9 days. Nothing means anything to somebody who's addicted to heroin after 9 days. You can offer them $1,000 cash on the table to come back in a month, or you can give them $10 right now. Everyone walks out of the room with $10. There's no valuation of the future. And the point of this is, I think, as we think about how to deploy our investments and our strategies, we have to think about the long term and the future. And I'll just summarize by saying what I hope I told you, maybe not all of this, but what I tried to get in was you need to start early. There's good biological basis for that. We need to have enrichment in many ways. It comes in many flavors. I didn't show you the data on this, but confidence, instilling confidence changes these circuits. Repetition is good. Concentration and attention to work is important. Social interactions facilitate learning. Spacing is important, not cramming, drinking from the fire hose. High stakes, a little bit of stress, that's okay. Too much stress is destructive. Learn by doing and get some rest. And finally, we can invest in our future now, or we can pay for our past later. Thank you very much.